Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. As you guys know, we're in the coronavirus shutdown, so these videos are intended for my students taking my human anatomy and physiology courses at Del Mar College in Corpus Christi. Now, this video itself is for my Human A&P 2 class, Biology 2402, and this is the fourth video in the series on the urinary system. We're going to try to wrap up the urinary system so we can start taking those quizzes next week. So, by the way, today is April 22nd. If you're following along, if you're in my class and you have your notes set, we're on page 92 in the second section. We finished with the regulation of the glomerular filtration rate. You got to understand the mechanisms by which we adjust the filtration rate. It's an important concept because the filtration rate affects how much toxins we get out of the body. It also has an effect on how many of the nutrients and ions we can reabsorb. We went over everything that's happening up in the glomerulus and the Bowman's capsule. By the way, I realized in the last video I said when fluid comes flying through the glomerulus, what I meant was this fluid's flying through the renal tubule. I think y'all may have caught that. But anyway, so we are on page 92. We're going to talk about tubular reabsorption. Tubular reabsorption is the ability to pull the nutrients and ions to transport them from the renal tubule into the peritubular fluid and then essentially back into the peritubular capillary so we can retain those substances. Anything not pulled out of the renal tubule will end up in the collecting duct, will end up in the minor calyx, the major calyx, and pretty much once things are in the collecting duct, it's next to impossible to recapture them because we don't have the transport proteins there. So now, when we talk about tubular reabsorption, the reabsorption of the nutrients, 100% of it occurs in the proximal convoluted tubule. And then some of the ions we reabsorb through the thick ascending limb. We get variable reabsorption of ions in the proximal convoluted tubule and some variable reabsorption at the thick ascending limb. And we reabsorb water here. So tubular reabsorption really involves this. And I've talked about this before, but I like to reiterate these things so that we really have a strong understanding of it. If I were to cut through the proximal convoluted tubule or the thick part of the loop of Henle, the reason it's thick is because the cells in here are simple cuboidal cells. I didn't draw that very well, but these simple cuboidal cells form a lumen in here. And that lumen is a specific size. And then there's little microvilli all along here, which is why they call these brush border cells, because the microvilli look like the hairs on a little paintbrush or something. Okay. Now, if I look at the thin part of the loop, the lumen is roughly the same size. I lack the microvilli, and the cells are simple squamous cells, so the walls are so much thinner for osmosis to occur. If I have water in here, and I have a whole lot of salt out here, if the salt cannot cross the membrane, then it will pull the water out, creating an osmotic effect. That's the osmolarity, is the concentration of the salt in a specific fluid. In this instance, we're talking about the peritubular fluid. As we go deeper in the loop of Henle, the osmolarity outside the tubule becomes higher and higher and higher, pulling more and more water out, essentially creating a very concentrated tubular fluid, which makes a concentrated urine. I pee out a little tiny bit to get all the toxins out. That's the whole point of this. So we're going to talk about tubular reabsorption. We're going to talk about the loop of Henle, and we're going to talk about how we regulate all of this stuff. Okay, so now, one of the things I want to point out is if I took one of those cuboidal cells, on one end there's all the microvilli, on the other end is called the basilar surface, and right outside the basilar surface, we're going to have a peritubular capillary. So, this would be the tubular fluid in here. As you know, we have nutrients and ions, like glucose and amino acids and sodium and potassium. And we have little transport proteins in the membrane of these microvilli. The whole point of microvilli is to increase the surface area, because if the top of this cell was smooth, and I tried to make the cell the same width as the original, I only have that much surface area. I can only have this many transport proteins, right? But if I increase the surface area, I can increase the number of transport proteins throughout here. And all those transport proteins can be helping transport these substances into the cell. And then down here, I'm going to have some transport proteins, although I don't need microvilli. We're going to pump some of those substances out. But at least we've recaptured them and we're holding them in the cell, and then we can pump them out into the peritubular capillary. So those microvilli are an important adaptation. These transport proteins are an important concept. Okay? So now, how do these transport proteins work? Now, we could talk about these specific ones, and there's a diagram in your textbook, and I know when I was a student, we had to try to memorize all this stuff, and it was, I was able to do it. A lot of students just got overwhelmed and gave up. As we moved up through our education to our junior and senior level and higher level physiology courses, these concepts actually became solidified and I understood them. So I'm going to take a lot of the specific details out. And I just want you to understand the concepts so that when you do read the textbook, these definitions, these words mean something to you, and then you can understand them. And then I'll throw maybe one or two at you just as an illustration. So one of the definitions you should know from early in part 1 is facilitated diffusion. Okay? As you guys should know, diffusion is the movement, the passive movement of a substance from an area of high concentration to an area of low concentration. I talk about this all the time in class because it's one of the most important concepts for healthcare professionals. When you're trying to administer certain fluids through an IV, you're affecting diffusion and the osmolarity of somebody's blood. You're affecting a lot of these concepts we're talking about. So if I have two areas separated by a semi-permeable membrane, I put a bunch of stuff. This could be albumin, this could be sodium, this could be potassium chloride, whatever it is, or glucose, or oxygen, whatever the substance is. If I put a bunch on one side and a little on the other, I've created a concentration gradient. And this substance naturally wants to flow from an area of high to low concentration. We call that passive movement diffusion. If this membrane is permeable to that stuff, it's going to move until it reaches an equilibrium. One of the natural laws of physics. By the way, our body does not want equilibrium, it wants a steady state, and that requires us to maintain this imbalance very often with energy. Now, in facilitated diffusion, let's say this substance is not capable of crossing that membrane, but on its own. But let's say I put another substance in here, and that substance, when it binds to this membrane, actually opens the hole a little bit more. So now, without the green stuff, let's say the purple stuff could not diffuse, but if I add the green stuff in and it starts moving across, I all of a sudden notice the purple stuff starts moving across too. So that means that the green stuff, whatever it may be, is aiding or facilitating or helping the other stuff diffuse. Without it, this won't diffuse. So the purple stuff needs the help. It needs to be facilitated. So that's facilitated diffusion. That is the passive movement of a substance that requires the presence of another substance to diffuse. The second substance facilitates or helps the first one. Okay? You need to look up that definition, but that's essentially what it is. It is the passive movement of a substance, it's diffusion, from an area of high to low concentration that requires the presence of another substance in order to move, okay, in order to diffuse. The next one is called active transport. We have beaten active transport, I think, to death in part 1 A&P, and I keep talking about it in part 2. But in active transport, this requires the expenditure of energy, ATP. Active transport simply means we're going to move a substance across a membrane, but we have to use energy to do so. We have to burn cellular energy, or ATP. It is an active process. This stuff might flow down its concentration gradient. If I want to move some of this against that flow, that would be like moving upstream. You have to burn energy to go against the current of things. So in active transport, we're simply burning energy, like the sodium-potassium pump and calcium pumps. Co-transport. So if I look at the term co-transport, you need to look up all these definitions and write them in. But co-transport is when two or more substances are going to move in the same direction. And I keep hearing some beeping, and I have no idea what that is. Anyway, so in co-transport, I have to have two substances that are going to move in the same direction. So if I have a semi-permeable membrane, and I have some ion channel here, and I open this ion channel, then these two substances are going to move in the same direction. They both can cross like this. Co meaning with, they transport with each other or together. The opposite of co-transport is counter-transport, and in counter-transport, what would happen is two or more substances move in opposite directions. So one substance is moving in one direction, the other substance is moving in the opposite direction. Okay? So counter-transport is simply two or more substances moving in opposite directions. I'm not going to write all that out. It's in the note set. Okay? So you need to know these four definitions. Facilitated diffusion means one substance can only move when there's the presence of a second substance that helps it. Active transport simply means it requires energy. And, facilitated diffusion is always a passive process, but there is some types of active transport. Excuse me. I apologize, but my dog has to go in and out all day, especially when I'm lecturing, and I'm not going to burn the first 10 minutes of this video. Co-transport is two substances moving in the same direction. Counter-transport is two substances moving in opposite directions. You need to understand those four definitions so that if you do read the textbook, when you get to higher levels of physiology, what we can say is that you'll read sentences in the book, and our book covers a lot of this, but I'm not going to test you on it. There's enough to learn already. But it may say at the luminal surface, these two substances, maybe sodium and glucose, are co-transported together by facilitated diffusion. Co-transport means they're going to move in the same direction. Facilitated diffusion means they require each other. So we can actually combine these. And we might say at the basilar surface, so maybe sodium and chloride are counter-transported in opposite directions. Okay? And it's an active counter-transport. That means we're going to burn energy to switch two molecules around. Okay, those aren't the best examples, but at least it's a way that we can talk about at the luminal surface. These two substances can be co-transported by facilitated diffusion, or they can be co-transported by active transport. They can be counter-transported by facilitated diffusion, or counter-transported by active transport, and then the same thing can happen at the basilar surface of the cell. And some parts of the cell, sodium and glucose move in the same direction. Other areas, bicarbonate and chloride are going to move in opposite directions, and you can read all of that and understand it. I'm not going to spend a lot of time making you memorize all those details. Okay, I want you to understand the concept. So now, when it comes to the next section, the last part of page 92, we have these two concepts. One is called tubular maximum, and one is called the renal threshold. And I like to think of it in these terms. Okay, so imagine if I had a conveyor belt here. I have a little wall, and then I have students standing on this side. Let's say I got 5 students over here, or 4. Okay. They got 2 hands. This conveyor belt's moving at a certain pace. And over here is a big giant dump truck or something. Okay? If I start to dump substances onto this conveyor belt and it's going to move in this direction, I can tell you guys, hey, whatever you see on there that you like and want, grab it. Whatever y'all collectively get, you pull off the conveyor belt. You guys can all divvy up and split up evenly. Whatever gets by and falls into the dump truck, you have lost. And I'm over here throwing stuff onto this conveyor belt. If I'm throwing laptops and iPhones and wads of cash and jewelry and things, y'all are going to be trying to pull that stuff like crazy. I could also throw little bags of dog poop and vomit and other trash and debris and nasty stuff on there. You don't want to touch that. So the good stuff The stuff you want to keep would be things like nutrients, glucose, amino acids, lipids, fatty acids, and glycerol. And there's waste, trash. Now, if this conveyor belt is going, and at a certain period of time, let's say in a 10-second period, which this thing can go by in 10 seconds, what if y'all could grab exactly, 12 things? And at any moment, there's 12 objects on there that y'all can grab. There might be more, there might be waste, but let's say at one moment I have 12 valuable objects on there. For 10 seconds, if I can throw 12 things on there, then you guys would pull all 12 objects off. You would get 100% reabsorption. These are the cells lining the proximal convoluted tubule, all of the entire tubule. Okay, this is filtration occurring at the glomerulus. We're dumping good stuff and waste onto the conveyor belt. This is going to be urine in the collecting duct, which eventually will be removed from the body. So there is a point, like I said, if this takes 10 seconds to run across, and within 10 seconds y'all have the ability to grab 12 things and keep them, then that is your tubular maximum. The tubular maximum, and this is written in my note set, you don't have to go look it up. The tubular maximum is the concentration at which we can reabsorb 100% of a substance. So there's a certain concentration that when glucose is going by here, and y'all should probably know this one. For glucose, the tubular maximum is approximately 180 milligrams per deciliter, tenth of a liter. Okay, so 180 milligrams per deciliter. So at any one time, if I have that concentration of glucose in the proximal convoluted tubule, I will reabsorb 100% of it. That's the maximum. If it's below that, I'm still going to get 100%. If I only threw 8 objects on here, y'all would gather all 8. Okay? So now, and that would be the good stuff. Obviously, you wouldn't touch the trash. It would go into the collecting duct. Now, what if at any one time I drop 12 valuable items on here plus one more? Let's say I drop this one extra thing on here, and within that 10-second period, that one extra thing starts to show up in urine. Then we call that the renal threshold. You have crossed that threshold. The renal threshold is the concentration at which any substance begins appearing in urine. So, for example, if I had 181 milligrams per deciliter, then I might have some sugar, just a trace amount, show up in urine. If you have way more sugar than this, when someone's blood sugar gets up above 200, 220, 300, 400, man, they're losing lots of sugar in their urine. There's something wrong. Usually some kind of indication that you have diabetes. So anyway, that's what they look at when they do those glucose tests with diabetes. They have you consume a certain amount of sugar. You have to be fasting. You drink that orange stuff, and then you come back the next day and you urinate, or within a few hours, depending on which test you're doing. But they can measure how much of the stuff, how much of that sugar that you consumed is ending up in urine, and they can see if you're filtering properly and reabsorbing properly. So the glomerular filtration rate also has an effect on this, because if I know what the tubular maximum is, but let's say I speed up this thing to where that stuff goes by in 2 seconds, well, then I'm going to have some of the valuable nutrients end up in urine because I can't recover them all. It's moving too fast for the transport molecules, for them to bind to the transport molecules and get transported. So if my blood pressure is so high that I'm filtering so rapidly, eventually some of the stuff makes it through here and I can lose some things in urine. Another thing that could happen is if I form enough capsular hydrostatic pressure, that's going to eventually build up and fight the glomerular filtration pressure, or the glomerular hydrostatic pressure, and I could shut down filtration also. I'm not going to ask you about those two details. I just want you to understand the concept. And this is the concept. Tubular maximum is the concentration at which I get 100% reabsorption. Anything below that, I'm going to absorb 100%. Renal threshold is the concentration at which it begins appearing in urine. Okay, you can think of it as tubular maximum plus one extra molecule. All right, so now we're done with page 92. If you're following along in my note set, we're going to move on to page 93, which is countercurrent multiplication. I already discussed this in a previous video, okay? But again, these concepts are so important, and there's so much of what you're going to do in nursing and other training programs that you guys, you really need to understand this concept pretty well. And I'll tell you one other thing. Over the years of teaching this, I've been teaching at Del Mar for 20 years. I taught at UTSA for a few years. But at Del Mar, I pull my A&P classes every semester. You guys that are in my class know this. I asked you on day one, how many of you are hoping to have a career in the health sciences? Usually between 98, 99, and 100% of my students are going for nursing, occupational therapy assistant, physical therapy assistant, respiratory therapy, dental hygiene, surgical technology, medical lab techs, what else? Diagnostic medical sonography, echocardiography, all of these programs. There's a couple of more that I left off. No insult to those programs. But we have, I think 11 or a dozen health science programs. We also have kinesiology majors that have to take A&P. And we also have EMT paramedics. They have to have it. Okay, all of these programs require this. An overwhelming majority of my students every semester, let's say I have a class of 60 students in a lecture hall, usually somewhere between 58 and 60 are health science majors, allied health science majors. Very few are biology majors. They're usually allied health science, kinesiology, or then there's one or two other programs that make students take this. Now, you guys have to understand these concepts. And the reason I'm telling you this part right now anyway is because I got an email a couple of semesters ago. And I try to reiterate this. Those of you who are in my class know that during the fall semester or during the semesters in part 1 and part 2, I try to point out this isn't about getting a score on a test and collecting points, okay? Because getting a grade might get you into the program, but if you don't understand the material, you will flunk out. You're not going to be able to make it through the program. So this is about learning and understanding and comprehending. You're collecting tools, intellectual tools in your tool belt so that one day you can utilize these tools in A&P to solve problems in your professional programs. Very often I have someone say, well, I'm in respiratory therapy, so I just need to get through this. Well, I got an email a few semesters ago, and I've shared this with many classes. I actually read the email. I don't divulge the individual. But the person told me, you know, Mr. Long, as we're going through the respiratory therapy program, I'm realizing how many of the other systems, digestion, urinary, and other systems impact our respiration and our breathing rate and how much our breathing impacts those systems' functionings. So we do adjust blood pH here. Metabolic acidosis and metabolic alkalosis occurs because of what's happening in the kidneys and our ability to rid the body of these things. But you also get respiratory acidosis and alkalosis, and you have to figure out, based on the pressures of oxygen and carbon dioxide, which one's which. There's a neat trick for doing that, problem solving, but I'll share that with y'all some other time. But nonetheless, you must understand these concepts. And the kidney is one of the ones that all the students come back and say, man, I didn't pay that much attention to urinary. I learned it, but I didn't realize how important it was and how many pharmaceuticals and how much impact it has on all the other systems. So make sure you learn this stuff. This is why I'm reiterating it. Anyway, enough said about that. Countercurrent multiplication, countercurrent exchange. If I were to cut out the loop of Henle, it looks kind of like this. I have the thick and thin descending limb and the thin and thick ascending limb. We're going to go down here. We're going to go up here. Okay. Now, I like to do this way so that you get to visualize it. Let's say I have water molecules represented in blue. There's 12, there's 15, there's 18. Let's say there's 21 of these. I got a certain number. I have nutrients. Not nutrients, because nutrients wouldn't be in here. Ions. I'm going to represent ions in purple. Sodium, potassium, and chloride. I have some ions dissolved in here. 1, 2, 3, 4, 5, 6. Okay? So let's say there's a certain number of those. and I have some waste molecules. All the amino groups and ammonium ions and urea and stuff. I have a certain concentration. It's diluted out by a lot of water. As that goes through the thin descending limb, if I start to pull all these water molecules out, by the time I get over here, let's say I have way less water molecules. If I have not pulled any of the ions out, then my ions are way more concentrated in here than they were before. I have the same number of salt molecules, I have less water. Now, that also means that my waste molecules are also going to be fairly concentrated. I don't want to lose that much salt. That's too high of a salt concentration. So as I go up the thick ascending limb, these simple cuboidal cells lining this have ion pumps that are going to pump some of these ions out. And as I pull some of the ions out, by the time I get to the top over here, I have this amount of water. I have a little bit of ions. And if I did this right, the ion concentration here, compared to the number of water molecules, would be equal to the ion concentration here, about 1/3. I think I have 21. I should have 7 molecules there, and I have 6 and 2. I may not have drawn it perfectly, but this concentration and that concentration have the same osmolarity, the same concentration of salt. But what's also happened is I have very dilute toxins here, and now I have a very concentrated urine. I'm getting rid of a lot of toxin, but only losing a little bit of salt and water. That's the whole point of the loop of Henle. The reason we call it counter-current multiplication is this. These two are flowing in opposite directions. That's a counter-current. Current is flow, counter is opposite. The multiplication is this. When I have a batch of fluid coming through here, if this batch of fluid is at this point and I pull all this salt out, then this gets to be very salty in the peritubular fluid. That's going to create an osmotic pull, so to speak, an osmotic pressure. When the next batch of fluid comes down, the salt that was pumped out by the ascending limb multiplies how much water is going to osmose out of the descending limb. And when that batch of water gets over here, now that is all multiplied how much salt I need to pump out. So this is going to multiply the amount of salt pulled out of that batch of water. As I pull the salt of water, as I pull the salt ions out or pump them out, then the next batch of water that comes through here in the tubular fluid is going to have this osmotic pull. So this multiplies how much water I pull out, which multiplies how much salt I have to pump out, which multiplies how much water I pull out of the next volume of fluid, which multiplies how much salt I pull out, which multiplies how much water. And you can see how it feeds itself in this constant cycle. Each branch is multiplying the effects of what is moved out of the opposite branch, and they're moving in opposite directions. That's countercurrent multiplication. All of that is explained to you in my note set. Finally, we're getting close to the end. There are a couple of concepts here. There's one called obligatory water reabsorption. In obligatory water reabsorption, if you look at it, obligatory comes from the word oblige. The same word as obligation. When you have an obligation, you have to do that. There's no choice. You are obliged to do so. Obligatory water reabsorption is what occurs between the proximal convoluted tubule and the loop of Henle to the beginning of here. All through the proximal convoluted tubule to the loop of Henle is obligatory. Remember, I reabsorb a lot of water in the proximal convoluted tubule. I have to do that to maintain body hydration. And then I reabsorb another 20 to 25% or so in the loop of Henle. I am obliged to do that. My body has to do that to stay hydrated. Now, what happens over here in the distal convoluted tubule is called facultative water reabsorption. The facultative water reabsorption is that of water reabsorption that is facilitated. Facultate, facilitate, they're the same words, to help out. In facultative water reabsorption, this is the water reabsorption that occurs in the distal convoluted tubule and is facilitated or helped out by the presence of two different hormones. ADH increases water reabsorption from the distal convoluted tubule into the peritubular capillaries, and aldosterone increases salt and water reabsorption from the distal convoluted tubule, and some experiments show in the part of the proximal portion of the collecting duct where the fluid would go in. So we can reabsorb a little bit of water and salt there under extreme conditions when we're dehydrated. If I don't take in enough fluid, or if I lose too much fluid, or I have really low blood pressure for an extended period of time, then this rate of flow through here is going to be low. And in the last video, and we talked about the hormones that affect the glomerular filtration. ADH targets the distal convoluted tubule, increases water reabsorption, and renin from the juxtaglomerular apparatus converts angiotensinogen from your liver into angiotensin 1, goes through the lungs, hits ACE, angiotensin converting enzyme, gets converted to angiotensin 2. That goes to the zona glomerulosa of the adrenal cortex and causes the release of aldosterone. Then aldosterone targets the distal convoluted tubule and increases salt and water reabsorption. So those are the obligatory and the facultative water reabsorption. Obligatory is the natural reabsorption that occurs in the proximal convoluted tubule and the loop of Henle. Facultative is what occurs in the distal convoluted tubule and parts of the collecting duct due to the presence of hormones. The hormones help it out. Okay. All of that is reviewed in my note set on bottom of page 93. So if you guys are in my class, do that. It also continues on page 94 and talks about all that. Now, just a few more concepts we have left to discuss, and some of this I'm not going to go into too much detail. Tubular secretion is when we secrete or add harmful substances from the distal convoluted tubule or from the blood into the distal convoluted tubule. We are secreting out of the body, and at the distal convoluted, we talked about this when we did the nephron physiology, I think in lecture 2. But tubular secretion is how we dump excess potassium, because we get abundance of potassium in our diets, and hydrogen ions and some ammonium ions are all dumped into the distal convoluted tubule so that we can get them out. There's a little reiteration in the note set about where the ammonia and the ammonium ions come from. You have to remember, as we break down proteins, the amino acids, if we need them for energy, we have to deaminate or remove the amino group. We combine that with hydrogen ions, and that forms ammonia. Another hydrogen ion can be pulled in due to the strong attraction from the structure of the nitrogen, and that forms the ammonium ion. All three of those are very, very toxic for you. And then there's also, we combine that with carbon dioxide and urea to form urea. All of those compounds are very harmful, and they all come from deamination of proteins. So protein metabolism, while it's necessary at times, can result in some harmful substances and toxins. Now, urinary transport, we simply move urine from the renal pelvis through the ureters to the bladder. When we look at the bladder, I'm going to draw this out for you. Let me erase some of this. When we look at the urinary bladder, you should know the anatomy of the urinary bladder. And if we look at the wall itself, the walls are a little thicker than most other organs anyway. And then we have a tube coming out called the urethra. And urine gets squirted out of your body through the urethra. Females have a rather short urethra because the bladder is sitting right where the vagina is, but males have a much longer urethra as it courses through the penis. But nonetheless, the urethra is there, and that's where urine comes out. Now, the inner walls of the bladder are actually folded when the bladder is empty. Those folds are called rugae, just like they are in the stomach. Ruga would be singular. So when the bladder is empty, it's all kind of folded on itself. As it fills up with fluid, those folds disappear and it can distend. So our bladder has some compliance to it. As we talked about before, compliance is the ability of an organ to change shape, to accommodate changes in volume. And the ureters would be draining in here from the kidneys and dumping urine into the bladder. Now, One of the unique structures of the bladder is this little structure that has 3 branches, and one of them might be coming up towards us. So I'm only going to draw 2 of the 3. But that thing is called the trigony. It looks like the word trigon, but it's a Greek word. It's pronounced trigony, kind of like the Greek play Antigone. If you've ever seen or read the play, it's a really cool play. Anyway, look it up. It's an interesting. I'm surprised no one's ever made a movie about it, but nonetheless, excuse me, I dropped my. The trigony has 3 branches, hence the term trigony. It's got baroreceptors or stretch receptors in it, actually, so that as the bladder distends, when the top of the bladder can't stretch anymore, the bottom of the bladder will start to stretch, and then that's going to send a signal into the very end of the spinal cord, the sacral region, and some of the sacral neurons will fire back and cause us to start contracting that smooth muscle. And as that smooth muscle contracts, it will start to compress the bladder, forcing the urine out. We also have 2 urinary sphincter muscles. Okay, there's a muscle that's kind of deeper inside of here, and then one closer to the opening near the end. The internal urethral sphincter is under subconscious control. That's autonomic. So not only will I start to compress that muscle, I'll start to relax the internal urethral sphincter. We don't have any control over it. The external urethral sphincter we have conscious control over, and then that's when you can go, uh-oh, I better hold this stuff in till I get somewhere where I'm not going to embarrass myself. I mean, if you're talking to someone you're attracted to and then you start wetting your pants, it's all over. Unless they're into that, I guess. I don't know. There's all sorts of stuff when we get to human reproduction. But nonetheless, so the sacral region of the spinal cord is receiving input and can reflexively fire back and cause us to relax the internal urethral sphincter and start to compress the detrusor muscle. The detrusor muscle is a really thick muscle layer here that surrounds the urinary bladder. And that detrusor muscle, when it compresses, is going to squeeze the bladder and help you urinate with a lot of force. So please know that the internal urethral sphincter is under autonomic control. The external urethral sphincter is under conscious control. It's a somatic muscle, skeletal muscle, so to speak, and we can choose to relax it or constrict it. And that's how you try to hold urine in for a while. Eventually the pressure will override it and you embarrass yourself. But nonetheless, or you warm the pool, or your inner tube if you're floating the river. Sorry. Know that the external urethral sphincter is under conscious control. By the way, one of the nerves that helps control that is called the pudendal nerve. We talked about this in part 1. It is part of the sacral plexus. The pudendal nerve, if we damage the sacral part of the spinal cord, or we damage the pudendal nerve, and some people say pudendal, we lose control of our bladder. And so people have to have a permanent catheter in. People who are paraplegics and quadriplegics, very often, if they have damage to this nerve, they can't control urination and defecation. So finally, the urethra is the tube that squirts or carries the urine to the external body. And then there's what we call the Micturition reflex, Micturition is urination. It's the urination reflex. We just covered that. So you need to read that section. Incontinence. Continence comes from the word of the ability to contain. If you are incontinent, you cannot contain your urine. So the inability to hold our urine in is called incontinence. That can happen due to the muscles wearing out and damage due to the muscles. As we get older, we become incontinent. Women who have lots of babies, as you know, the stretching of the uterus can compress the bladder and stretch things out. It can damage the ability of the bladder. And sometimes the bladder can collapse on itself and not hold a lot of urine. There's a surgical procedure. When I worked in surgery, we did a few times called the Marchetti procedure, where they resuspend the bladder for females that have incontinence. Anyway, if y'all are surgical techs, y'all will learn all of that. So that should conclude all of the notes on the urinary system, all right? So we are done. We're going to be taking a test pretty soon over all these concepts. If you're in my class, there'll be some worksheets posted with these videos on my Canvas webpage or my Canvas page from Del Mar. If you're watching these videos in YouTube land, I hope you glean something from them. I hope they help you in some way. Again, these videos are not the way that I want them to be. These are very quick and impromptu. My dog barks, my kids walk by, I get distracted and start fumbling over my words. Please forgive me. I'm doing the best I can with what I got. I hope to very soon be able to redo all these videos when we're back to normal so that I have the time to edit them and make them nice and slick and put better diagrams and graphs and stuff up for you guys. In the meantime, this is what we got to work with. Thanks for watching. I hope you learned something. I hope you had as much fun as I did. I always have a blast talking about the human body. So see you guys on the flip side for the last round for lecture text 6. See ya.